Hello and welcome students. Today we are discussing an interesting topic which is viruses. It includes influenza virus, retroviruses, HIV, viroids, virusoids and prions. Before discussing viruses in detail, let's first know its objectives. First one is influenza virus. Second one is human immunodeficiency virus, that is HIV. Third one is viroids. Fourth one is prions. And fifth one is virusoids. Now let's discuss the first objective, that is influenza viruses. Influenza is a highly infectious viral illness. The name influenza originated in 15th century in Italy from an epidemic attributed to influenza of the stars. Influenza typically causes fever, headaches, muscle aches and more significant cough. However, mild cases of influenza are similar to colds. Of the two stereotypes, influenza A occurs more frequently and is more dangerous. Although most epidemics are caused by influenza A, both A and B stereotypes frequently co-circulate during yearly outbreaks. Although influenza B is usually less severe. Influenza viruses are RNA viruses in the family of Orthomyxoviridae, which includes 4 genera: influenza A, influenza B, influenza C and Hogotoviruses. They are enveloped negative-stranded RNA viruses that can be distinguished on the basis of antigenic differences in the nucleocapsid and matrix proteins. Influenza A viruses are roughly spherical with glycoprotein spikes on the surface and genome consisting of 8 RNA fragments that encode 10 proteins. The hemagglutinin and neuraminidase and matrix proteins are embedded in the envelope lipid bilayer derived from the host cell. The matrix protein underlining the envelope is the major determinant of virion morphology. The nucleoprotein associates with each RNA segment to form the ribonucleoprotein complex, which also contains small amount of 3 polymerase subunits. Hemagglutinin is responsible for the virus attachment to the cell surface, binding to the sialic acid residues in cell membrane glycoproteins, thus triggering the viral fusion and its entry. The proteolytic cleavage of hemagglutinin by serine proteases present in the infected tissue exposes hydrophobic fusion domains that mediate membrane fusion. Neuraminidase cleaves terminal sialic acid from glycoconjugates present on respiratory mucins, cells and progeny virions. This action is the key of importance for the release of the virus from infected cell and consequently for spreading infection throughout respiratory tract. Now, diagnosis of influenza viruses. In most individuals, self-diagnosis on the basis of clinical symptoms is adequate. Rapid antigen tests are currently the diagnostic tests of choice for influenza. These tests are performed directly on the patient samples and can be highly sensitive and specific. PCR is now available for influenza, although it is not yet in widespread. Treatment of influenza. While most people require only rest and fluids, some people will benefit from specific antiviral agents. Amantadine and rimantadine are primary systematic amines which interfere with the viral uncoating by blocking the action of influenza A's M2 protein. These drugs are only effective against influenza A as influenza B lacks M2 protein. They are also very effective at preventing infection if given during influenza outbreak. Now coming to the second objective, that is HIV, Human Immunodeficiency Virus. AIDS is an illness that damages a person's ability to fight off disease, leaving the body open to attack from ordinarily harmless infections and some forms of cancers. AIDS is caused by the virus called HIV. This virus infects certain types of white blood cells, primarily CD4 cells, also called helper cells or T4 cells, and monocytes. HIV stands for Human Immunodeficiency Virus. Once someone is infected with HIV, the virus will remain in their body for the rest of their life. There is currently no cure for HIV and no vaccine to prevent people from becoming infected. However, treatment can help most of the people with HIV to live much longer and feel well. The structure and the life cycle of HIV. HIV is an enveloped RNA virus. As HIV buds out of the host cell during replication, it acquires a phospholipid envelope. Protruding from the envelope are PEG-like structures that the viral RNA encodes. Each PEG consists of 3 or 4 GP41 glycoproteins capped with 3 or 4 GP120 glycoproteins. Inside the envelope, our blood-shaped nucleocapsid of the virus is composed of protein and surrounds 2 single strands of RNA. 3 enzymes important to the virus life cycle are reverse transcriptase, integrase, and protease are within the nucleocapsid. HIV is a member of the group of the viruses known as retroviruses, which share a unique life cycle. Once HIV binds to a host cell, the viral envelope fuses with the cell membrane and the virus RNA and the enzymes enter the cytoplasm. HIV, like other retroviruses, contain an enzyme called reverse transcriptase. This allows the single-stranded RNA of the virus to be copied and double-stranded DNA to be generated. The enzyme integrase then facilitates the integration of the viral DNA into the cellular chromosome. Provirus is replicated along with the chromosome when the cell divides. The integration of provirus into the host DNA provides the inactivity that enables the virus to avoid host responses so effectively. Production of viral proteins and RNA takes place when provirus is transcribed. Viral proteins are then assembled using the host cell's protein-making machinery. The virus protease enzymes allows for processing of the newly translated polypeptides into the proteins, which are then ultimately assembled into the viral particles. The virus eventually buds out of the cell. A cell infected with the retrovirus does not necessarily lyse the cell when the viral replication takes place. Rather, many viral particles can bud out of the cell over the course of time. HIV transmission. HIV can be transmitted from an infected person to another through blood, semen, vaginal secretions, breast milk. Blood contains the highest concentration of virus, followed by the semen, followed by vaginal fluids, followed by the breast milk. Activities that allow HIV transmission. First one is unprotected sexual contact, direct blood contact including injection drug needles, blood transfusion accidents in the healthcare settings or certain blood products. Mother to baby, before or during birth or through breast milk. Sexual intercourse, sharing injection needles. An injection needle can pass blood directly from one person's bloodstream to another. It's a very effective way to transmit a blood-borne virus. Sharing needles is considered a high-risk practice. Mother to a child, it is possible for an HIV-infected mother to pass the virus directly before or during birth or through the breast milk. Breast milk contains HIV, and while small amount of the breast milk do not pose significant threat to an infection to adults, it is viable means of transmission to infants. Symptoms of HIV. Many people who are living with HIV have no obvious signs and symptoms at all. Recent evidences show that between 70 to 90% of people who become infected with HIV experience flu-like symptoms within a few weeks after infection. The most common symptoms are a fever, a rash, and a severe sore throat, all occurring at the same time. These symptoms in an otherwise healthy person may indicate recent HIV infection. Large lymph nodes or swollen glands that may enlarge for more than 3 months. These symptoms are also seen First one is frequent fevers and sweats, skin rashes that does not go away, short-term memory loss, slow growth or frequent illness in children, cough and shortness of breath, difficult or painful swallowing, confusion and forgetfulness, nausea, stomach cramps and diarrhea, vomiting, vision loss, unexplained weight loss, unusual tiredness, severe headaches. Now, treatment for HIV and AIDS. Blood tests for the presence of T cells. According to the Centers for Disease Control, patients with less than 200 T cells per cubic millimeter of the blood have AIDS. With proper treatment, most infected patients can lead relatively normal lives for many years. Even with the onset of AIDS, symptoms can be greatly diminished by the treatment. Antiviral therapy, known as antiretroviral drugs, slow the process of HIV because fewer HIV cells are formed. Now coming to the third objective, that is viroids. Viroids are small infectious pieces of RNA that were first discovered in 1971. Viroids are smaller than any viruses and do not have capsid. They also differ from viruses in that their RNA does not code for any protein. Viroids are plant pathogens that can quickly destroy entire fields of citrus, potatoes, tomatoes, coconut palms, and other crop plants. The first viroid discovered is potato spindle tuber viroid, PSTV. It was discovered by Danier in 1971. Viroids are notorious for causing plant disease of considerable economic importance. The induced symptoms depend largely on the host plant and are usually characterized by diminished growth, stunning, leaf epinasty, necrosis, and flower and fruit deformations. Now coming to the fourth objective, that is prions. Stanley first proposed the term prion in early 1980s. He found evidences of neurological disease caused by agents that appeared resistant to the process that normally destroy the nucleic acids. The idea won the Nobel Prize in Physiology in 1997. A prion is an infectious protein which is imagined to infect tissues and reproduce first by changing its own conformation and secondly by inducing other kinds of protein to do the same. Prions or proteinaceous infectious particles cause a number of rare diseases in mammals. Prions are abnormally shaped proteins found in the brain and the nervous tissues of infected animals. When those tissues are eaten by another animal, the prions enter the animal's bloodstream and go to its brain. In the infected animal brain, the prion interact with the normally shaped proteins, causing those proteins to change the shape and become abnormal and infectious. The brain of affected animals are full of spongy holes. Scurvy is a fatal degenerative disease that affects the nervous system of the sheep and goats. Scurvy is one of the several transmissible spongiform encephalopathies, which are related to bovine spongiform encephalopathy and chronic wasting disease of deer. Like other spongiform encephalopathies, scurvy is caused by a prion. Scurvy has been known since 1732 and does not appear to be transmissible to humans. Now coming to the fifth objective, that is virusoid. Virusoid is an infectious agent that infects plant in conjugation with an assistant virus. Virusoid genome are 220 to 388 nucleotides long. A virusoid genome does not code for any proteins but instead serves only to replicate itself. Virusoid can replicate in the cytoplasm and possess a ribozyme activity. RNA replication is similar to that of the viroids, but each requires that the cell to be infected with a specific helper virus. An example of a helper virus is the subterranean clover mottle virus, which is associated virusoid. Virus enzyme may aid replication of the virusoid RNA. The virusoid is incorporated into the virus particle and transmitted as a satellite, a separate nucleic acid not a part of viral chromosome. Replication of the helper virus is independent of the virusoid. Virusoid belongs to the large group of infectious agents called satellite RNAs, which are found in the bacteria, plants, fungi, invertebrates, and vertebrates. Some satellite RNAs encode proteins that are called satellite viruses, and like virusoid must co-infect with the helper virus in order to replicate. One satellite RNA infecting humans is hepatitis delta virusoid. It has a circular single-stranded RNA genome of 1700 nucleotides. Its helper is the hepatitis B virus, which is associated with the liver disease. Co-infection with both agents results in a more severe infection. It's all about this topic. Hope you understood it well. Thank you.